My name is Adil, and my name is Adil Basha, and today I'm going to be talking about PT-POP and how you can use photochemistry for the potential diagnosis of tumors in the human body. This research was actually performed in collaboration with Sindhu Shankar. So I actually did this research in collaboration with Sindhu Shankar and Dr. Shrika in the UNT Department of Chemistry. Before I begin, I just wanted to go ahead and give you an overview and the clinical significance of photochemistry and PT-POP. Photochemistry has become a promising source of biosensors. And to give you some context on what it is, photochemistry is essentially the usage of light to react with molecules, and these molecules in turn would create a Wickham catalyst for new reactions or new compounds could be formed. So it's a really revolutionizing stuff. And people have already begun to take advantage of the properties of photochemistry as a science, and they've made some really interesting technologies with it. In fact, the U.S. Department of Energy has actually ended up testing these biosensors on to check photosynthesis in plants, and also to study the visual system of the human eye. So we've already begun to use biosensors like PT-POP in actual biological mediums. And in fact, UNT's very own Dr. Nisa Satumtir and Dr. Sophia Kinyanjui have both already done prior testing on PT-POP, and it sheds some light on its ability with regards to detecting oxygen levels in actual biological mediums. In fact, I think Dr. Sophia Kinyanjui had tested with the C. elegans worm, and she found some very promising results. And that actually prompted us to go ahead and test PT-POP further. But before I actually talk about what we learned from PT-POP, I wanted to give you an understanding of its properties and why it's so important to us. So what is PT-POP? PT-POP is a phosphorescent binuclear molecular system that absorbs light to function as a photocatalyst. So when you think of catalysts, you think of something that can promote a reaction. Well, PT-POP works like that, but with regards to what we're studying, we actually look at its oxygen sensitivity properties, and we mostly focus on its emission of green light. And I'll actually get into that a little bit later. As of right now, what we know is that the molecule is very unstable, and its optimal conditions in the biological medium have yet to be understood. And so the purpose of this experiment was to actually find the right temperature, the right solvents, and to see when its emission would be at its max. Because by having a high emission, an emission of light, which means that it shines very brightly, that means that we can see very clearly what we want to see. Like, for example, with tumors, if we have a brighter emission, we can pinpoint exactly where the cancer is. We can see where things are off in our testing. And so what we know right now is that it's a highly luminescent molecule, it's oxygen sensitive, and it's water soluble. And you can see the diagrams on the left-hand side. The figure A is actually a PT-POP in a two-dimensional form, and you can see platinum makes up its backbone. Figure B is PT-POP in powder form, and that's in ambient lighting. And figure C is PT-POP when it's dissolved in water, and UV light is actually being shined on PT-POP. And you can see that it's emitting a greenish glow. And that's actually very important in the context of our studies. So I'll get into that a few minutes later. So before we began actually stabilizing PT-POP, what we actually did was we wanted to test its effects on the effects that solvents and pH actually had on the PT-POP molecule. And so what we ended up doing was we got phosphate buffers for the solvent for the pH testing, and these phosphate buffers were prepared with different pHs, ranging from 5.7 to 8.1. And when we ran it through a photoluminescence machine, we ended up finding that the PT-POP, when it was dissolved in a pH of 6.1 phosphate buffer, would have the highest emission intensity. That emission intensity would be around 4.5 times 10 to the fifth AU. So that just shows that it shines brightest when it's in a pH 6.1 phosphate buffer. And that's actually very important when in context of biological testing, because many human tumors actually fall near that pH range. So if we did want to implement this in the human body, it would be pretty effective because the PT-POP molecule would be able to function inside of a tumor's microenvironment. And the other test we did was for the effect of solvents on PT-POP's emission intensity. And so we tested a variety of solvents ranging from water, methanol, DMSO, and a mixture of all of those. And we ended up finding that the peak intensity would actually be highest. The emission peak would be right here. It would be highest when it was actually a mixture of water, methanol, and DMSO. So all three of them combined. And if you look down here, you'll see the emissions when all of these solvents, when PT-POP was dissolved in these solvents and placed under UV lighting, you'll see the how they shine very differently. Like water right here has a very dim emission, whereas methanol, DMSO, and water, when they're all combined, actually have the brightest emission. So that just shows, to put it in the context of which solvents PT-POP is very compatible with. And so I mentioned earlier that PT-POP is still very unstable as of right now. And so we ended up doing is we conducted some polymerization studies. So what we did was we had to first synthesize the PT-POP monomer. And so what we did was we had PT-POP stock molecule, which we bought, and we had allyl chloride. And when we combined them together, there were some byproducts that were formed, such as potassium hydroxide, water, and chlorine. And what ended up happening was, in the end, we ended up forming PT-POP allyl chloride monomer. And from this monomer, we had to begin the polymerization process. So we had a three-step process. We had initiation, propagation, and termination. And this is similar to what happens with radicals. And that's actually true because we're using radicals in this case. So for the initiation step, we did UV laser radiation of a molecule called 1-hydroxycyclohexylphenylketone. And that broke up the molecule into free radicals. And now these free radicals ended up combining with the PT-POP allyl chloride monomer we had synthesized earlier. And after a series of steps, it ended up terminating the reaction, and we had a non-cross-linked PT-POP allyl chloride polymer. So now they were all they're woven together into a long polymer, but they were not cross-linked. So we ended up adding a cross-linker into the solution, and we created a cross-linked PT-POP allyl chloride polymer. That was the end product of our polymerization studies. And now we wanted to actually test to see if the luminescence of the emission of PT-POP, the monomer and the polymer, stayed consistent. So we didn't want to sacrifice the emission wavelength that we already knew, and like the emission intensity that we had known from the original PT-POP molecule. So that was the case here. We actually didn't sacrifice anything. We were able to stabilize the molecule without having to change the actual chemical properties of PT-POP at all. Because you can see right here, the emission when it's exposed to UV light is still the same throughout. It's green here, it's green here, and it's green here. So it shows that nothing fundamentally changed within the PT-POP molecule. So after that, we actually wanted to do some oxygen sensitivity testing of the PT-POP polymer. And this is actually very important because this is vital to application in the human body for cancer, for the purpose of studying cancer. And we ended up finding here through a series of nitrogen and oxygen purging studies, we found that it was a reversible sensor, meaning that upon exposure to nitrogen, upon exposure to oxygen, its emission would significantly reduce. So if you think of a really bright green light, when it's exposed to oxygen, the PT-POP's green emission would be much, much dimmer. But when it's re-exposed to nitrogen, after 5 minutes, it'll actually return to its original emission state. And that's important because if we do use PT-POP as a diagnostic tool and we want to reuse it, it's actually if we find a way to salvage it, it's actually very useful because PT-POP is made of platinum, and platinum is a very expensive metal to get a hold of. So it's important that we have a reusable sensor. It cuts costs of manufacturing the PT-POP polymer, and it makes it a much more viable tool for detecting cancer or for detecting anything that involves oxygen. So after that, we wanted to actually test to see if PT-POP was more stable than the PT-POP polymer was more stable than the original PT-POP molecule, and that was the case. If you see on the left-hand side of the screen in graph A, this is thermogravimetric analysis of PT-POP. The y-axis shows the weight of the molecule, and the x-axis shows temperature. So as the temperature increased, we'd see that the molecule began to rapidly decompose because the bonds were being broken apart by the high temperature. And around 86.76 degrees Celsius, it rapidly it just decreased linearly, but at around 500 degrees, it was 84% of its original weight. On the other hand, if we see the PT-POP polymer in graph B, we'll notice that it maintained its molecular weight percentage up to 315 degrees Celsius, and then it began to exponentially decompose, and it reached 20% of its original weight after around hitting 500 degrees Celsius. So it shows that although at very high temperatures it decomposes faster, it still manages to maintain its integrity, its molecular integrity, up until 315 degrees Celsius. And we use that property of the polymer to actually go into further testing to test its how its emission is reacts to exposure to heat. So that's actually led into our spectral studies. So here we compared the PT-POP polymer at room temperature and the PT-POP polymer at 260 degrees Celsius. You'll notice how I said back here that 315 degrees Celsius was a threshold before the molecule began to lose its integrity. So this is well within its threshold. And what we ended up finding here was when it was heated 260 degrees Celsius for 30 minutes, it actually ended up having a red emission when it's exposed to a wavelength of 368 nanometers, its excitation wavelength. And I'll get into that in a few minutes. So you can notably see a change in its emission from green emission to red emission when it's heated for 30 minutes. So this led to our UV-vis absorption studies. So to provide some context, the original PT-POP molecule that we're working with, that Dr. Necessa Tomtira and Dr. Sophia Kinyon-Joy worked with, that molecule, its excitation wavelength and its emission wavelength, which is the light emission, was 515 nanometers. So when you look at a photoluminescence machine, you have a laser. You have to program the laser to shine a particular wavelength of light. That's its excitation wavelength. So the wavelength is 368 nanometers. And when the light shines, it hits mirror at a 90-degree angle, and it goes into the molecule, which is being held in a little compartment. And in turn, the molecule actually absorbs the light and then emits its own wavelength of light, which is traditionally 515 nanometers. And if you look at the visible light spectrum, that's in the green area. That's where the color green is. So here we did a reverse study, essentially, where for UV-vis absorption studies, we know what the emission wavelength is, but we want to find the excitation wavelength. And here we ended up finding the excitation wavelength was around 368 nanometers. And for the heated sample, the excitation wavelength was also 368 nanometers. So nothing had really changed with regards to the excitation. So now we had to do photoluminescence studies because things had changed, obviously. We noticed a red emission. We wanted to test to see what the wavelength of that emission was. And what we ended up finding for the PT-POP and ambient temperatures, when it was excited at 368 nanometers, it ended up having an emission wavelength at 515 nanometers, which is right here. So we still see the same green emission, the same green emission for PT-POP in solution, along with all the other excitation peaks for the green emission. But for the heated PT-POP sample, what actually ends up happening is the green emission is killed by the heat. And you'll see here that it's around 2.0 times 10 to the 5 AU, whereas over here, its green emission is actually 4 times 10 to the 6 AU. So there's a very notable decrease that can be noticed right here. But there is a new red emission, a broad red emission that's dominating. That's actually formed at around 638 nanometers right here, and at corresponding excitation wavelength. So we have new properties that are associated with this heated PT-POP sample. And this is actually, if you look at the visible light spectrum, red is usually around 600, the 600 nanometer area. So this is a red emission, and that's what we saw in the previous slides. We saw a picture of PT-POP emitting red light. So now we wanted to do oxygen sensitivity studies because that's essentially the foundation of this experiment, is to make sure that it can be compatible with testing cancer. So what we ended up doing was we redid the oxygen sensitivity study for the PT-POP polymer, and we found that nothing had really changed. The nitrogen exposure to nitrogen would increase the wavelength, the emission intensity, whereas exposure to oxygen would kill the emission intensity, and it was reversible. On the other hand, for the heated PT-POP sample, we found that the green emission was reversible. So at 515 nanometers, the green emission, when it was exposed to nitrogen, it would go up, and when it was exposed to oxygen, the intensity would go down. But for the red emission, actually, what ended up happening is after multiple runs with nitrogen and oxygen purging, it would slowly start to kill the emission until it was no longer reusable. So here, the red emission, after a certain period of usage, it can no longer be used. So for the purpose of actually detecting cancer with a reusable sensor, this is not viable. The red emission is not viable at all. But the green emission can still be used, but it is very, very low compared to the green emission of a non-heated sample. So as of right now, we have not actually found the purpose of the red emission, but we are very confident that it can't be used in biological mediums for right now, just because of how incompatible it is with regards to constant testing. So moving forward, our future plans for PT-POP involve the continuation of heating studies and further exploration of PT-POP's red emission for industrial use. So we've known that PT-POP's red emission might not work with biological mediums, so we're actually looking to use it for industrial purposes because you have to heat this molecule up to 260 degrees Celsius, and it's only viable for such a short period of time, I think 24 hours before it starts to degrade naturally. So this isn't really useful in the context of clinical testing. We also want to use nanodots and encapsulate the PT-POP to create more effective and safer delivery systems. Because I told earlier that PT-POP is actually founded on platinum, on the platinum element. And platinum and a lot of other metals are very toxic to humans. So we actually need to find a way to isolate them from the body and only interact with cancer cells that are in the body, which leads me into the next plan we have, which is cytotoxicity studies to determine if PT-POP actually has the means of differentiating between healthy human tissue and cancer cells. And so that's very important actually in the context of clinical studies because we need to make sure that PT-POP can function on its own and it doesn't indiscriminately start killing cells that are healthy. That could actually be very dangerous for humans that may have cancer. So that concludes my presentation. I just wanted to go ahead and give some acknowledgments to Ms. Sindhu Shankar, who was my graduate student and who was the one that was heading this project. My friend Naman Basin, who was a research colleague of mine, who also worked in my lab and helped me with some of these studies. Dr. Muhammad Omari, Dr. Shrika Marpu, Dr. Nisa Satumtir, and Dr. Sophia Kinyanjoy. Dr. Satumtir and Dr. Kinyanjoy were both pioneers in this field. They paved the way for Sindhu and I to actually continue doing the PT-POP studies. So without them, we wouldn't have had a basis for where we had to start. That concludes my presentation. If you have any questions, feel free to ask.
Well, thank you, Adil. Okay, Adil, while other people might be thinking about it, let me get to something kind of fundamental would work as a method to identify cancerous locations in your body. If I understand what you're doing correctly, it sounds to me like the trick here is the difference in fluorescence that PT-POP will have depending on the amount of oxygen
That's right.
around it. And so that would imply that you would just have that in the bloodstream or something like that, and then you would just see where it is fluorescent.
That's right. So
a little bit.
There's a testing in current medicine called fluoroscopy, where you inject a dye into the human bloodstream, and what ends up happening is it runs through a scan, and you can the human body lights up, right? So that's kind of what we're trying to implement it as. It might not necessarily be the end product of what we're making, but essentially we inject the PT-POP. Of course, it has to be encapsulated because of the fact that it's toxic to humans. So we'd have to still do some further stability testing to make sure that it can differentiate between tumors and human cells. What ends up happening is the PT-POP would have a lower oxygen emission, so a lower green light emission when it's within a tumor's low oxygen microenvironment, because we know that tumors have lower oxygen levels. Higher oxygen levels because of the fact that they have higher metabolism. They have to take in more of our resources, and that's what makes them cancerous, because they take our nutrients, they take our blood gases, and they use it for themselves to grow. So these tumors are going to have higher oxygen environments, whereas the rest of the body will have moderate levels of oxygen, of O2 saturation. So when we actually end up injecting this PT-POP into the bloodstream, the PT-POP molecules that are within the tumor microenvironments will actually glow dimmer compared to the PT-POP that's in the rest of the bloodstream. So we'll see pockets of dimmer areas all spread across the human body where the tumors might be located. And that helps us pinpoint exactly where they are, how big they are, how extensive they've grown, if there's any metastasis. So that's what I would hope to implement this as, but that might not necessarily be the end product.
Yeah, it sounds almost like as far as where fluorescence or phosphorescence is going to occur, it seems to be almost the opposite of green fluorescent protein, which you might know is something that was had become very popular in the last 15 years. A Nobel Prize was given for that. And in those cases, the effort was to get green fluorescent proteins that would attach to the cancerous cells. And you would be able to identify the location of the tumors by having that region shine, as it were.
Mm-hmm.
In your case, do you worry about the fact that detecting the points of darkness might be a little bit more difficult for instruments? And how does that compare with green fluorescent proteins? If you have run into that, and you don't have to have.
We haven't actually tested it this far, but I would assume that modern day instrumentation it would have an issue differentiating between dimmer areas, like you said. But if you notice the PT pop emission intensity, it can vary greatly. I think in my slides I had mentioned that in certain conditions it can become much lower. Let me just find that. So yeah, we have tested in varying oxygen levels actually, and what we ended up finding was that when PT pop is exposed to around like 70% O2 saturation, its emission would be higher compared to 100% O2 saturation. So in our purging studies, we actually exposed it to varying percentages of gases, and we ended up varying percentage of oxygen gas, and it did make a noticeable change with regards to intensity. So I think the issue with any instrumentation would just be how sensitive it is to varying levels of luminescence. But I feel like that could be very easily fixed. It would just we just have to do some testing on these instruments, but that's not really in our field of expertise. It's more along the lines of like biological engineers. So we would have to work with them to make sure that we have a certain threshold for what the computers can detect during the scanning.
I guess I was muted.
Yeah.
Thank you, Adil. I appreciate your presentation. And I guess I had a rough start with my time management bumping around. But if others have further questions, we'll save those for the very end of the session. We will go ahead and move on to the next speaker at this time. And Shrika Eddula is our next speaker. And Shrika is going to give us a presentation titled Mitigating the Effects of Hazardous Organic Solvents by Using LFERs to Develop a Novel Model to Guide Solvent Selection. So Shrika, you can take it from here. Thank you.
Okay, wonderful. Well, yeah, so my name is Shrika. I'm from the Texas Academy of Mathematics and Science, and today I'm going to be presenting to you the novel model that I created with the help of my research lab to mitigate the effects of these hazardous organic solvents on the environment. So before we get too far into the scientific methodology, I want to show you this picture here that I have of Lake Onondaga. And Lake Onondaga is located in Syracuse, New York, upstate New York. And it's actually been noted as one of the most toxic places to live in the United States. And this is due to the fact that municipal companies have been unloading their industrial waste and municipal sewage into this lake as a means of disposal. And this repeated action for over a century has caused this lake to be listed as one of the most toxic places in the world to live in. Unfortunately, Lake Onondaga is only one of the many places worldwide which have succumbed to the same fate. And here I have a recent image taken from the Environmental Protection Agency that shows the various hazard rankings of various compounds. And you'll see that some of these compounds are actually noted as highly hazardous and even toxic, while some are noted as safe to use. And so, but what I ultimately hope to do through this project is identify these compounds that are hazardous and replace them with these safer ones. So once again, the chemical industry uses over 1 million metric tons of solvents every single year. And these are crucial to the chemical manufacturing industry as they're used in a variety of means. But once again, these are disposed into the environment, leading to both irreversible environmental damage, as well as threats to worker safety, as these workers that work in these industries are constantly exposed to these materials on a daily basis for 8 hours a day. And this severely threatens their emotional and physical well-being. If you can recall, I'm pretty sure many of you have seen the mesothelioma victims and disclosures on TV that have been released in the past few years. And this is just another case study of those. So thus, what we need to do is we need to find a method in which we can gather safer solvent alternatives and effectively replace these toxic compounds. Okay, so once again, my ultimate goal was to determine a model in which we could quantify the chemical properties of all these compounds. And so I broke this down into 3 further main points. So number 1, I wanted to develop this model. And then number 2, I wanted to be able to extend my measured data to not only predict the chemical behavior of the compounds that I've studied, but also various other compounds as they're being discovered. And then lastly, I wanted to ensure that we can apply this to the environmental aspect, so that way we could mitigate these effects. So I broke down my research into 2 phases, and the first phase involved developing the model. And so before I get too far into the model, I want to describe to you this idea of solute-solvent interaction. So we see in the bottom right, the red ball is actually going to be our solute, while the other gray balls are going to be your solvent. So what happens when these 2 interact is that the fundamental characteristics of both the solute and the solvent determine how the interaction will take place. So some of these characteristics include, but aren't limited to, the ionization energy, the bond strength, the charge, and the hydrogen bond associated with these solute-solvent molecules. So if we can understand why these interactions take place and how these interactions take place, then we can effectively predict how any solute-solvent interaction will take place. So unfortunately, there's very little solubilizing data out there that actually gives us chemical data for these chemicals. And additionally, there has yet to be a condensed format in which researchers can use this to effectively find these alternative compounds. So thus, when I went about making this model, I chose the properties that were most important to understanding the solvation process. Of course, chemicals have a diverse array of properties, and we can't study every single one of them, but I'll get to this later, but I chose the 6 main ones that were the most important. And this was based off the concept of linear free energy. And what linear free energy is essentially a chemical principle that correlates the kinetic energy, the Gibbs free energy of the system with its kinetic rate. And then so essentially what this allows us to do is create these 2 equations. And these 2 equations are each describe a different state of solvent-solute interaction. So our first one actually describes 2 condensed phases, which would be a solid and a solid. And our second one describes the solute transfer between a gas and a condensed phase. And on the left-hand side, we have our dependent variable. And this is going to be our log K and our log P values. And essentially, this is what we're going to be measuring through our experimental data, or training the model with. And then on the right-hand side, if you recall, if you see the capital letters, those are going to be our independent variables, and those are actually going to be our solute descriptors. And in each of these experimental trials, that's what we're going to be altering. And then lastly, the lowercase letters that you see are what we're going to be determining in the end. And I like to think of this as similar to a social security number, but instead, in this case, it's actually for a chemical. So our goal is to ultimately determine a unique social security or set of solvent descriptors for each compound. So once again, I mentioned this before, I decided to focus on 6 main solute-solvent descriptors that model these interactions, and these are given here. Essentially, each one of these is a unique characteristic. For instance, E will tell you the molar refraction of the given solute. So this basically just means that if you have your compound and it interacts with the wavelength of light, how is it going to react? We have S, which is our electrostatic polarity, and this is essentially just its polarizability. When there is a dipole in its place, what is going to happen? What is it going to do in reaction to this dipole? A and B, respectively, are hydrogen bond donating and hydrogen bond accepting. Those are pretty basic, just how likely and how able it is to give and receive a hydrogen bond. V is its McGowan volume. And this is actually similar to like density, except it quantifies specifies 3D space by using the atomic size and the chemical bond numbers of the molecule. And then lastly, L, this descriptor only exists in the gas phase because it's a logarithm of the gas to hexadeciane decane partition coefficient. And basically this was just going to tell you at 298.15 Kelvin, how much is this gas going to disperse. So now that we had a model, I went on to phase 2 of my experimental methodology, which included training this model with experimental data that we collected. So before we went into collecting our experimental data, we had to choose our compounds of interest. So in order to choose my solvent, I read some literature and I saw that a group of green compounds, as noted by the Environmental Protection Agency, alkyl carbonates, have been noted to be very low ecotoxicity and very good at biodegrading. And however, these haven't been very well represented in previous studies. And then so I thought that through the study, this would be a perfect way of bringing to light the environmental capabilities of these molecules. And how these are formed are with a carbonic acid and an alkali base. And frequently these, in the past, these have been used in lithium ion batteries and the removal of carboxylic acids, as well as polymer membrane preparation. So you can see that these are very versatile. They're used in a wide array of settings, and we hope to expand upon these through the study. So now we had to choose our solutes. And to choose our solutes, what we essentially wanted to do was ensure that we had a wide array of chemical diversity, because that would allow us to have a metric for a chemical on this end of the spectrum and this end of the spectrum. And then we could predict the chemical properties of all the solutes in between. So to do that, we looked for solutes with a large range of solute descriptor values. And eventually what we settled on, this was done with the help of past literature, is we settled on 24 organic compounds and 26 inorganic compounds for a total of 50 total solutes. So these are the two equations that I had mentioned previously. And we see that once again, the capital letters are going to be our independent variables, and they're going to exist for each the gas phase and the condensed phase. And ultimately our goal through this experimental methodology is to train these two equations to calculate the lowercase letters or our solvent descriptors. So for our experimental methodology, if you recall, our dependent variables are the log P and log K values, and that is what we're going to be calculating through this study. And so how to do that, first we gathered a 100 milliliter flask, and in this flask we put the organic solute as well as 2-propanol. And the reason we used 2-propanol was basically because it's the cheapest diluting agent that we could find that not only didn't react with these compounds, but also diluted them effectively. And then after that, we conducted a series of dilutions on this flask. Okay, so yeah, and then we had dilutions from 1 through 10 to all the way through 9 through 10. And the point of this was to use the principles of the Beer-Lambert law. So what we did is we used a spectrometer to calculate the absorbance values of each of these dilutions. And the wavelength that we use is predetermined using a spectrophotometer in order to ensure that at this wavelength of light, DEC nor the solute, DEC, which was our solvent diethyl carbonate, did not absorb any light. This way, any absorption of light could be attributed to the solutes. So once we had these epsilon values, absorbance values, we were able to determine epsilon values using the Beer-Lambert law. And if you're not familiar with the Beer-Lambert law, essentially what it is a linear regression model that shows the shows the relationship between absorbance and the molar absorbant coefficient epsilon. And this allows us to determine the molar concentration at an unknown absorbance and vice versa. So in order to actually test our samples, we had static equilibration methods used to ensure that the solvent and solute reached a saturated state. This was just to ensure that all the interactions that I mentioned before had taken place. And so to do this, we left these in an amber glass bottle in a water bath that you can see in the image on the left. And we usually left these for around 3 to 4 days, and we would check every single day to ensure that there was no solid or crystallization at the bottom, because that would mean that it was oversaturated. And additionally, if there was no particles floating around, that means our solute solution was unsaturated and we had to add more solute to it. And if you recall earlier, we had the Beer-Lambert law. So using that Beer-Lambert law, we were able to perform dilutions on these samples until their absorbance were found to be in the range of the standards. And because we had the equations generated with the Beer-Lambert law, we were able to use our linear regression plots to determine the molar concentrations. We performed 24 solute solvent trials, 4 to 6 trials for each, just for accuracy and precision. And then using these molar concentrations that we determined in the last slide, we were able to change these to mole fraction solubilities using the sample mass, the molar mass, the dilutions that we performed, as well as the volume of the flask, which was 100 milliliters. So this is the, if you recall earlier, the dependent variable that I mentioned that we were calculating. And on the far right-hand side, you'll see the CS organic, which is essentially the numerator of our log P and our log K values. And this is the chemical formula that we use to calculate this value using our mole fraction solubilities. And so now that we had that, we have the molar solubilities for both the gas and the solid phase. When we compare this to water just as like a standard, we now have those known values, and we can go on to create 50 simultaneous linear equations. And if you recall earlier, the reason for the 50 is because we had 50 solutes. So we had 50 solute solvent interactions taking place, and thus 50 linear equations to depict these interactions. And just as you would solve a very simple XY linear system of equations, we solved these using the IBM SPSS statistical software. And so these are the log K and the log P values that were determined for our solute of solvent in this case, which was a diethyl carbonate and alkyl carbonate. And you'll see here, these numbers won't mean much right now, but essentially what we've done is that we've created a unique set of these solute descriptors for this compound. And when we performed regression analysis using our statistical software, we found that our data actually had a R-squared value of 0.994 and 0.999 respectively, indicating that there was a very strong correlation between our experimental data and after we collected these, what we were able to do is, using chemical databases, is compare these social security number or solute descriptors to the solute descriptors of various other compounds. And this was taken from a chemical database that we had access to. And once again, this was done very similar to the way you would do it in your algebra class. You have two XY coordinates, and so you would use the Euclidean distance formula to calculate the distance between these. Except in this case, since we had 5 descriptors, we used 5 variables. So the formula can be seen there. And the calculated solute descriptors were actually compared to these 4, or these are the 4 hazardous compounds that we found the distance to be the smallest to. So we have here 2 nitriles, an acetate, and dibutyl ether. And from the very first thing that I brought up was that the Environmental Protection Agency actually notes all four of these compounds to be hazardous. So because we have shown that these compounds are similar in behavior to our environmental green diethyl carbonate, we're actually able to replace these in the chemical industry setting, and that way we can mitigate the environmental impact of that these hazardous compounds have. And then lastly, to ensure that our model was accurate, we compared our experimental results to some theoretical figures taken from literature, and we found that our results were within 0.13 log units of all of the theoretical measurements, and this indicates that we had a very high precision and were very accurate in our experimental measures. So in conclusion, we accomplished the 3 main goals of our study, which was to devise a novel model in which hazardous compounds could be replaced with the safer ones. We've established a method in which solute-solvent interactions can be quantified. And lastly, we've been able to apply this to the environmental setting. So some future applications. In this case, we did only use the temperature of 298.15 Kelvin, which is standard temperature and pressure. but we have yet to study how these compounds will behave at other temperatures. Although we expect them to perform in a similar relationship, we actually want to confirm this. So some further testing would include testing them at various other temperatures. Furthermore, in this study, we've only focused on the environmental implications of this model, but we've seen that we could also apply this to the pharmaceutical industry, as every single year, millions of pharmaceutical drugs are declared safe and cannot not to be used. So if we were to apply this model there, we could create safer and more effective drugs, as we're able to predict the properties of the various drug molecules that pharmaceutical industries are attempting to use. And then lastly, we hope to further our investigation of various other groups of compounds. In this case, we investigated alkyl carbonates, which have been proven to be environmentally safe, but we also look to expand to various other environmental compounds so that we could create a chemical database in which we have a large array of replacement options for these industries. Lastly, just some acknowledgements. I want to thank my mentor, Dr. William Akery and Dr. Michael Abraham, Dr. DeBan for finding me this project to work on over the summer, and TAMS and the Honors College. Thank you. I'll be happy to take any questions.
Well, thank you, Shrika. So, Shrika, let me ask you something. Your presentation suggests that one of the main benefits will be replacing the specific solvents we use for different purposes, thus decreasing pollution in the future, or at least decreasing harm from that pollution. Do you envision solubility properties also having creating an opportunity for developing new methodology for cleaning up the sites that are already polluted?
Right, so in the lake that I brought up in the very beginning, so many chemical manufacturing industries have tried to reverse the damages that they've inflicted on the lake, but it's rather difficult to actually do this because the lake is so big. So what I've seen is that it takes a lot more resources to reverse this impact than it is to stop it overall. So what we hope to do through this project is just to stop the problem at its source rather than going at it after it happens.
Okay, fair enough. Well, thank you, Shrika. We will go on now to our next speaker, and Melody Nowlan is the next presenter, and her presentation is entitled K and X Variation in Tagalog.
Hi, my name is Melody Nowlan. I'm in the Department of Linguistics, College of Information. This whole topic is going to be switching gears from what we've been talking about. This is my research that I've been doing in linguistics. So it's K and X variation in Tagalog. I'm going to start off by saying that, languages are always changing continuously. Like, just think of right now, we're speaking, English, but go back 2,000 years ago and it wouldn't be the same. If we could time travel, we wouldn't be speaking the same language. So another example of language always changing is what's called the Great Vowel Shift. This occurred in early modern English, where vowels were raised. That means where they were pronounced in your mouth, you just sort of close your mouth a little bit, and that just makes the sound sound like it's higher. So for example, we have this long E, the E with a macron, that used to be pronounced eh, but because of the vowel raising, it's now E. So this word B-E-E-T, the vegetable, I think it's a vegetable. Anyways, it used to be pronounced bed, but now it's beat. We call them beats. So on to the language I'll be talking about. It's called Tagalog. If you look on the map on the left, this is a map of the Philippines where Tagalog is spoken. The shaded red areas is where Tagalog is. Tagalog is the whole language, and then there are 8 different dialects spoken all in those regions. So there is variation in Tagalog, just as there is in English. Variation just means you have one phoneme or sound. That's what the K in the, they're not parentheses, the slash K slash, that is a phoneme. And it can be pronounced in 2 ways. So the first K, the one with the slashes, that's the phonetic transcription, or sorry, the phonemic transcription, and then the phonetic transcription are the more, it just means it's more like specific to what the sound actually is. So the phoneme K can be pronounced as a K, as what the K is in the brackets, or a H, that's what the X is. So the K in the brackets, that's technically called a voiceless velar stop. It's a stop because all the airflow in your mouth stops. And then the X is called a voiceless velar fricative. And a fricative is just any consonant where there's continuous airflow. So think of an S, you pronounce S, and there's constant air. But if you say the sound T, your tongue is stopping that air. So T would be a stop, and S, the fricative. So that's a little background on that. An example word where you can see the variation is ako. Ako means I or me. And there are 2 possible pronunciations for this word. You can say it can be said with the stop, ako, or with the fricative, aho. And at the bottom, we have the consonant table and the vowel chart for all the sounds that appear in Tagalog. You can see the K in the velar and stop, if you go along where that is. And then it's voiceless, which is the first column if there are 2 consonants in the same spot. Okay. So these are my research questions. The first is the K and X variation in Tagalog restricted to certain phonological environments? That just means when K sounds like a K or when it sounds like a H, does that appear in certain environments? That means like in certain places in the word. And then if yes, what are the conditions of this restriction? So that would be me defining what parts of the word where K would be a H or a K. Okay, so here's some background in the fields. So for linguistic analysis, specifically acoustic analysis, a lot of people nowadays are using software to help them do this. One such software is called PROT, where you can look at spectrograms. So spectrograms let you visualize the sound. So on the right, we have an example spectrogram, and the transcriptions are right below each one. So the first one would be phi. The second one would be thigh and psi and then shy. So if there's a stop, then there would be no sound at all, right? Because there's no airflow, there's no sound being produced. There isn't an example of a stop in this spectrogram. These are all different fricatives. So the F, the theta, the S, and the esh, if you pronounce those, there's continuous air coming out. That's why it looks like there's white noise there. If it's a stop, since there's no noise, it would be clear. So on to Tagalog. So Tagalog phonology is a relatively new field. There's a lot of disagreement in it since it's so new and the research has been sort of incomplete. Generally, people agree that there are 16 consonants and 5 vowels. That's what I showed you earlier in the consonant and vowel charts. Some people, though, they do disagree with that. So Berger in 1967 argued that there were 16 consonants, 3 to 5 vowels, and 2 diphthongs. A diphthong is a vowel that has 2 parts. So like the sound I, it starts with A and goes into E. That's a diphthong. He doesn't explain, though, what the 3 to 5 vowels would be. So that was, that's a hole in his research. And then Himmelman in 2005 claims that there are 3 additional consonants in Tagalog. So that's F. That C is actually called a palatal stop. So your tongue would be flat on the top of your palate. Sorry, not a, yeah, that's a palatal stop. And then what looks like an upside down or rotated F, that's also a palatal stop, but that's voiced. The C is voiceless. So Himmelman thinks that those 3 sounds exist, but in like loanwords or in certain colloquial forms. But even until now, people well, not universally, more like most people agree that there are just the 16 consonants and 5 vowels that I showed you earlier. So these linguists, they do talk about variation, well, some of them. The variation is limited to vowels though, so it's not really, they don't really delve into consonants, how consonants can have variation. So that's the common theme with all of these researchers. And my hypothesis is that, first of all, the K and the X have to be variants of the phoneme K. Next, the phoneme K will tend to surface as the X intervocalically, or in between 2 vowels, and the stop K in other environments. And this is based on something called spirantization. It's where stops become fricatives between 2 vowels. So like that word that I showed you in the beginning, ako, the A and O, those are vowels, and the K is in between them. So that's why it's possible, or that's why I believe it's possible that people can say ako, because that K is in between those 2 vowels. So here's how I did my analysis. I picked this Tagalog television show called The One That Got Away from 2018. I used only the first episode from season 1, just so that, because there's enough data anyway, and also it would be a lot to do the entire season. So here above, I have screenshots of Prat, just on specific sounds. So in the spectrogram, if the K is pronounced as the stop K, then you'll see a break in the sound production. It's pretty noisy, I guess, in the spectrogram because it's a TV show, there's background noise, but for the most part, it's pretty clear to see what if it's a stop or a fricative. So if you look on the far left spectrogram, you'll see that there is a break in sound where the K is pronounced. Then over in the middle spectrogram, that one is an X, and you can tell because there's all that white noise. If there were a break there where there's no sound, then that would be a stop. But there's that white noise, so we know that it's the fricative. And then in the last spectrogram on the far right, you can see that it's a KX because it starts off with no sound, and then there's white noise immediately following that. So that would be the KX pronounced together. This is the rest of the methodology. So in total, there were 563 occurrences of the phoneme K. I had to throw out 22 because of just they were unusable data. Like, for example, if there was a word where K appeared right next to an H, I saw that it tended to be pronounced as the fricative X, the H, but it's hard to say if that was truly the K being pronounced as the H, or if it was the K assimilating to the position of the H, because if you say it quickly, it might come out as sounding like an X. So I had to throw those out. So in the end, I had 541 usable occurrences of K, and I calculated all the environments. So where the K showed up, word initial if it was in the beginning of the word final if it's at the end, intervocalic if it's between 2 vowels, and then a consonant cluster if there is a consonant that appears right next to the phoneme K. And then I also have what I call sub-environments, so pre-vocalic and post-vocalic. So for example, a consonant cluster. If you have, you can have a consonant cluster that happens before a vowel or after a vowel. So those sub-environments just make it more specific. And I'll show you later on too, I have different graphs. Here, this one is just showing the total surface forms of K. So that means the phoneme K, it's showing us how many times it showed up as the K or the H or the KX together. In some cases, it was actually deleted. I wasn't expecting that. But not that often though. It was only less than 4% of the data. Turns out KX together is even less than that, not even 1%. But X is over 20% of the data, and then K is over 75% of the data. So K, the stop K is still more common than all of the other sounds, but we still do have X occurring. And then here, these are the 4 main environments. So consonant cluster, intervocalic, word final, and word initial. When the phoneme K occurs in a consonant cluster, that's the top left graph, it always surfaces as a K, because none of the other options, the KX, the deletion, or the X, occur at all. In intervocalic position, there's a little more variation. It looks like the K and the X are about equal. K is still a little, the stop K is a little more common than the fricative, but the fricative is still up there at almost 17%. In word final position, there was only one occurrence of the X appearing. Every other time, it was only the stop K. And then in word initial position, it was still mostly the stop K, but X is also, it was also a possibility in that position. So I wanted to make it a little more specific. Here are the sub-environments. So on the left, we have where it appears word initially. So I split it up to where nothing was preceding the word. This would mean like in the very beginning of any utterance. It could be a sentence or phrase. The word that started with K was the very first word. So nothing would be before that. That's nothing preceding. There's no occurrences of the deletion, but a little bit of the X, which was interesting for me. And then I'll explain why in a minute. But it's mostly K, the stop K, that occurs when nothing is preceding the word initial environment. Post-consonantal just means that you have the word that starts with the phoneme K, and the word immediately before that ends in a consonant. So when you're speaking quickly, it's like the phoneme K is appearing in a consonant cluster environment. This is what that would be like. So here we see that no X appears. The fricative does not appear. It's most likely the stop that would appear, and then it's deleted sometimes, but not often at all. It was only one occurrence. And then post-vocalic just means that the word that starts with K comes right after a word that ends in a vowel. So this would be similar to an intervocalic position. And here, this is where it would be more likely that the fricative appears because it's in between two vowels. And then the word final environment, we have similar sub-environments. Instead of nothing preceding, it's nothing following. So that would mean that the word that ends in the phoneme K is the final word in whatever sentence or phrase someone is saying. And then in that case, it's only K, the stop K, I mean. In a pre-consonantal environment, that would mean that there's a word that ends in K, and then a word immediately after that that starts in a consonant. So this, again, would be similar to a consonant cluster. here, it's mostly the stop K that appears, and then one occurrence of the fricative X. And then prevocalic position, or prevocalic word final position, would be the word ends in K, and then there's a word after that that starts with a vowel, so that's like intervocalic. Here, only the stop K occurs. So what this means is, I have two possible rules. And the first one is that in a consonant cluster, including word initial post-consonantal position and word final pre-consonantal position, the phoneme K will most likely surface as the stop K. And in an intervocalic position, including word initial post-vocalic position, the phoneme K may surface as the fricative X. Okay, so for implications, this research gives us further insight into Tagalog phonology. As I said earlier in the beginning, a lot of the research in Tagalog phonology is relatively new, and there were a lot of holes regarding variation in consonants. This would be important in language acquisition. So if someone were to learn Tagalog, it'd be helpful for them to know about this variation, so that they could sound more like a native speaker and assimilate more into Tagalog culture if that's what they desired. It would also be useful for computational linguistics. So I'm sure you guys know about Siri or Google Home, just something you can, a machine you can talk to and it talks back to you. Well, if someone speaks Tagalog, tries to talk to it, and they have this K and X variation, then it's possible that their machine might not recognize what they're saying if they pronounce the X, the fricative, instead of the stop. So refining that code would be more beneficial to people trying to use this technology. But suggestions that I have for further research are to use clean voice recordings, not a TV show like I did. It would be optimal to use your own recordings that you could go out and take elicitations from people and then upload those voice files to Prot. And it would also be good to have a larger data set, and obviously give yourself more time to do the research. I only had this one semester, that's why I had such a small data set, and I couldn't go and make my own recordings, so this would be good for future research. These are my references, and these are my acknowledgements. Dr. Dina, Dr. Constantia, my capstone professor, Dr. Alexander Smith, my advisor, my supervisor for this, UNT, Department of Linguistics, College of Information, and obviously the Honors College for hosting Scholars Day, and of course the UNT Provost. Thank you guys, that's my presentation.
Well, thank you, Melody. I appreciate you presenting those results to us today. And we are almost really absolutely out of time. I know some have a commitment at 5 o'clock, but what about any questions for Melody? Does anyone have a question for her?
This is Hadge Ross from Linguistics. Hi, Melody. I'm glad to hear you're doing this intense work on phonetics. I have to go very quickly, but I wanted to ask, are there any other voiceless consonants that show fricativization intervocalically? Like, does T ever show up as either theta intervocalically or maybe S? Does P ever show up either as F or phi? So send me the reply by email if you would, because I have a class starting in 3 minutes. But
of course,
on your work.
Yeah, thanks so much. I will email that to you.
Okay, thanks a lot. Bye-bye.
Well, thank you for the question, and please do stay in touch as more questions come up. And for an ending of our session, why don't we just offer some virtual applause? We cannot see everyone, but I am putting my gesture on the screen, and thank you to the three presenters today. Appreciate that. Great job. And if you're not graduating, I hope you will present again and give us updates or further projects for Scholars Day next year. Take care now.